Okay, growth of a plant towards light is called phototropism. Okay, sugar is transported in the phloem. To carry out any tropism, plants must make cells on one side of the stem longer than the other. The main hormone to cause this would be auxins. Often plant cells need to replace older damaged cells. To do this, they would use cytokinins. A vine is climbing up a fence using small tendrils. This is an example of thigmotropism. Okay, in late September, deciduous trees, ones that lose their leaves, secrete something to signal a time of dormancy. Abscisic acid. Abscisic acid. Okay, number 7, the gas that ripens fruit, ethylene. Layer of the leaf where water is lost and gases are exchanged. No, spongy layer. Okay, that's where all that stuff is held. It goes through the stomates, okay, but the layer in which it happens is the spongy layer. Okay, what structure on the leaf controls the loss of water and exchange of gases? Stomates, or guard cells if they wrote that. Okay, and what are the two main functions of the stem? Transport and support. If they wrote conduction, I'd also accept that. Okay, you look at their answer. I mean, if they meant transport or they meant support with what they wrote, but they wrote it a little differently. I'll leave that up to you. No, yes. No, let's make it out of 11. Make it out of 11. Okay, so 1 mark for transport, 1 mark for support out of 11, guys. Okay, so give them a mark out of 11, let them see it, put it in the box. All right, so what we're going to be looking at here with the Transport and Plants Lab, and I hope it's going to work. Walmart didn't have the plants I usually get for this, and no one else did either, so I had to get the next best thing. So I hope it's going to work. We don't have exactly the plant I usually use for this lab. Okay. So the problem we're investigating here is scientists marvel at the ability of plants to raise water many meters above the ground, particularly since it is done with no apparent expenditure of energy. Okay. What are the forces that move water through plants? In this activity, you'll investigate this question. So the problem we're investigating is basically how do plants move water. We kind of already know the answer to that, but we want to make sure that that's the case. Okay? So what we're doing in this lab is we're going to alter the plant. Okay? We think right now that the way plants draw water up the plant is by what? Is by osmosis. And that force is generated where? In the leaves. Okay? Now, that's what we think. That's what it is, but we want to confirm that. So what we need to do is alter the plant in such a way that any other places where that transporting force could be generated are removed. Okay? Because a lot of people say, oh, well, the roots pump water up the plant, or there's, this or that goes on. I mean, we know that it's osmosis that does the job, but we want to prove it. So what we're going to do is we're going to cut pieces of the plant off, and we're going to stick them in water with no roots. Okay, so that's going to make it so that only the leaves can generate a pulling force that will draw the water up the plant. Okay, so we're manipulating the plant a little bit there in the way I kind of described. All right, so that's what we got to keep in mind there. We're watching for a couple of things. The observations we want to make are as follows. We want to make some qualitative observations about the appearance of the plant. Okay, what I mean by that is you're going to take pictures before, during, after, okay, the experiment. We'll take some pictures this morning. That'll be our before pictures. We'll take some pictures tomorrow. Okay, that'll be our kind of during pictures. And then we'll take a final picture on Monday. Hopefully the plants aren't dead by then. That'll be our kind of final pictures of the plants. Those will be our qualitative observations, and we'll look at them and see, check for the movement of the dye, okay, and see where the food coloring ends up in the leaves and in the plant and things like that. So those will be some of the things we'll be watching for. And we're also going to measure how much water actually moves through the plant by recording how much water we start with, and then at the end, recording how much we have at the end. Right? So we're really watching for a change in water volume, and we're watching for changes in the appearance of the plant. Okay? Those things are going to indicate whether water has been transported or not. Cam, you had a question? I have a plan for that. What I'll talk about when I talk about the procedure. But that's a good point. That's why I have a plan. Okay, for our hypothesis, okay, we want to talk about the forces involved in the transport of water in plants. So that's probably then what our if part should talk about. Okay, the if part should probably say this is what we think is the way that plants transport water. Okay, the and part will describe the experiment. The then part's going to have to talk about what you think your predicted observations are. Okay, and we said the observations we want to make are about the appearance of the plant and the volume of the water that it will consume. Okay, so you should say something about that in the then part. Okay, so for the procedure, there's a couple of things we have to do carefully. It's not a difficult procedure, but we do have to follow it quite to the letter. So we're going to set up our apparatus so that it looks I got to get to it. Well, it won't look like this because I don't have any of this plant, which is called Dieffenbachia. Okay, they didn't have any. I like it because it's got big leaves and they're white, so you can actually see the food coloring move into them fairly easily. But they didn't have those. In the end, your end product is going to be the plant in a graduated cylinder. Okay, and the graduated cylinder will be covered. So when we get in there, you're going to take two graduated cylinders. A large one, like 100 mil, and a small one, a 25 mil. Okay, you're going to mix your food coloring water mixture in the large graduated cylinder. Okay, fill it so that you've got 100 milliliters of water. Add 2 or 3 drops of food coloring is all you're going to need. Okay, 2 or 3 drops will be lots. Then you're going to pour that mixture into the smaller graduated cylinder. Okay, the reason we have to measure it in the big graduated cylinder is we're going to fill the little graduated cylinder almost to the top, and the measurement markings only go to about here on the graduated cylinder. So it'll be impossible for us to know how much is in there if we just pour into that cylinder without measuring. Okay, so what we're going to do is pour into that cylinder until it's close to the top. Let's say you've got maybe about a centimeter or a centimeter and a half clearance. Okay, from the top, because when you put your plant in, that's going to push some of the water level up. Okay, if you have it right to the top, it'll squirt water out. Okay, so that's what we'll do, and then we'll look at the 100 ml cylinder after we've poured water out of it and look at how much is left. Okay, so if we started with 100 and there's 45 left, how much did we use? 55. Okay, we used 55 mils. So that'll be all right. Our starting volume is 55 milliliters. It probably won't be that much because these are 25 mil grad cylinders. I think the most you can put in there past the measurement markings is like 35. Okay, so that's what we'll do so that we know the exact amount of water we start with. Okay, that's the first step. Once you have that ready, then I'm going to give you the finger off of a glove, and you're going to cover the top of the graduated cylinder with it. That's going to overcome the evaporation problem Cameron mentioned. Okay, so we're going to cover that up, and then you're just going to poke a hole in it with your pencil. Okay, just a small hole. We don't want a big hole. We just want it big enough that the plant can get through. Okay, and it makes a reasonable seal. Okay, around the plant. It's not going to be airtight, not going to be watertight, but it's going to be the best we can do. It's better than a completely open graduated cylinder. Okay, then you'll have this setup. Okay, now, sorry, you won't have this setup because you haven't cut the plant yet. Cutting the plant. The way you cut the plant is the most important part of the lab. You need to cut the plant underwater because if you cut it above water, air will get into the xylem, and that will wreck the cohesion, adhesion, hydrogen bond situation that's going on in there. So you're going to submerge it, cut it underwater, and cover up the cut end with your thumb. Okay, so you'll cover up the cut end with your thumb. Okay, cover up the cut end with your thumb and take it over to your apparatus. That way, no air gets into the xylem on the way over. Okay, when you get to your apparatus, you very quickly take your thumb off and put it in the water. Put it through the hole into the water, okay, and that'll be good. We shouldn't get any air into the xylem doing that. The problem with, carrying it over with nothing over the end is that we'll get air in it on the way over. So just cover it up with your thumb, and when you get there, quickly put it into the apparatus. Okay, then you're completely set up. All right, all you need to do then is take a piece of masking tape, write your names on it, wrap it around the graduated cylinder, and leave it in the window. Okay, and the setup for the lab is done at that point. Does that make sense to everybody? Okay, now, should you take pictures of it before you leave the lab? Yes, pictures you should take. Top of the leaves, bottom of the leaves, and the whole plant in general. Okay, and don't forget to record your amount of water that you start with. You need that starting volume area. No, I'm not going to worry about size and magnification, but I will want you to probably like point out things like, here's a, on the first ones, there's not going to be anything to label. It's the plant, and there's nothing wrong with it at this point. Tomorrow, you might be starting to see dye moving into the things. You might want to label, dye has gotten this far. On Monday, if you see spots on the bottom of the leaves and things like that, you want to label that. Okay, so the plant we're using is ivy, and it looked like Walmart hasn't watered them for a while, so I got the best ones I could find. Some of them still look sick. So they have some white edges. The leaves are fairly pale. All right, so we should be able to see the movement of water. And I think the stems are also quite pale, so we should be able to see the movement of the dye into them as well. Okay, so take pictures of the top as well as the bottom of the leaf, because we're going to see mostly evidence of movement of water in the bottom of the leaf, because that's where it's going to come out. All right, everybody follow me there? Okay, so I'll give you a couple minutes to work on just that pre-lab stuff, and then we'll head on over. Hopefully by then all the picture retake people will be back. Okay, incidentally, something else I forgot to mention. There are several choices for food coloring. When you get over there, I've got some blue, and I've got some red. I have green. Should you use it? Probably not. I used to just put it out there to see how many people would use it and not say anything. I'm older and more mature now, and it's not quite as funny as it used to be. So don't use the green. I'm actually going to take it off the counter so that you don't mistakenly use it. Okay? Because it just, for some reason, it's really difficult to see it as it moves through the plant. I can't imagine why. Yeah, people just don't think sometimes. So you can use the red or the blue, or you can put 1 drop of red and 2 drops of blue, or vice versa, and make purple if you want. It doesn't really matter to me. Okay, as long as you use one of those 2 colors so that it actually shows up in there. Okay, just before we go over to the lab here, I just want to quickly talk about the analysis, which obviously you won't be doing until next week. So the analysis questions that we need to answer here, we need to calculate the amount of water taken up by the plant. That's basically final volume, or initial volume minus final volume. It's not difficult. Okay. Then you need to explain any differences. They're going to be small. The plants don't typically move a lot of water, but it's enough to be measurable most of the time. Okay. Compare the rate of transpiration in your experiment with that of other groups in the class, and then explain any differences. So, you want to look at things like how big was the piece of plant that you used versus theirs. What did yours look like when you started? All that kind of stuff. For number 2, what evidence do you have that water followed the path discussed in class? Okay, so what we talked about, how water moves through the plant, is that what your observations say? Explain why or why not. Okay, and then thirdly, predict the effect of adding salt to the water and describe how you could test that prediction. Right, so in that, we're saying like we water the plant with salt water, or we put the plant in salt water instead of in fresh water. What's going to happen to it? Is it going to transport better, worse, why? Okay, the why would be the most important part there. Okay, explaining why it would not be, or why it would be, is whatever you feel. Okay, and then conclusion and evaluation, same as always, and we'll talk about the class discussion after I mark it. Okay, questions before we go over there? Okay, doors open. Off you go.